go today. And Dr. Rupali, I was seeing at her profile, she did some excellent work during her academic career, PhD, postdoc, and she was assistant professor. So then she changed her field from being from academics to that in the industry and a little bit away, more from the management viewpoint. So we will listen from her what was her journey and how to enjoy that success during the journey. So with this, I will hand over to Dr. Manju to continue.
Yeah, thank you, sir. And I will say words of thanks for Professor Ramakrishna, as well, as he always encourages such programs. And if as long as you are logistic with him, you are working on a logical idea and can explain things, you can't expect to hear a no from him. So he's always there for us. So thank you, sir. Yeah. Let me go to the. So all of you can see the slide.
Yes, ma'am. Yes, ma'am.
Yeah. So on this note, I formally welcome our today's second speaker of the series, The Possibilities for a Science Enthusiast. We have with us Dr. Rupali Gandhi, and I would like to take this privilege to introduce her with a top to bottom approach, so that students can relate with every bit and piece of this event, in a deeper sense. So Dr. Rupali did her BSc Microbiology from Delhi University. She pursued her master's in fermentation and microbial technology from Guru Nanak Dev University from Punjab itself. And there onwards, she went ahead with her PhD with the major area of work in the field of immunology at the National Institute of Immunology in affiliation with Jamia Millia Islamia. And it is from this time that I got to know her and is sort of associated with her. During her PhD, she worked on the autoantibodies in context of systemic lupus erythematosus with excellent work profile and the project and the way I've seen her working. She went ahead with her postdoc at Brigham and Women's Hospital, Harvard Medical School, and working in the area of autoimmune diseases, specifically the neuroimmune diseases, I would say. And from there onward, she has worked on different positions as a instructor to many more science scholars in an international setting. And alongside, she had experience on the managerial part of a laboratory. So with these all rigorous academic research and management based experiences, she was all prepared with the gusto to go ahead for an assistant professor position at Harvard Medical School at Brigham and Women's Hospital, Department of Neurology. She has shown her scientific expertise in the field of immunology, autoimmune diseases, and neurological diseases. And with this kind of academic profile and research output, which any one of us, would love to have, she made a major move towards moving into a healthcare-based industry system. And at present, she is working as a medical director at Sanofi Genzyme USA. So with this, I would just, knowing these details, I went back to the Sanofi Genzyme site, and this is what I saw. So I thought I would just share it with the students with the perspective that the Sanofi Genzymes, the motto is central to patients and science, at the center of everything they are doing. So that itself speaks a lot. And the rest we have with us Dr. Rupali Gandhi, who will, acquaint us and make us familiar with what all that goes on behind such a successful, journey the journey part has to be emphasized here. To reach the pinnacle of scientific excellence, I would say. Yeah. So Rupali, the stage is all yours. Thank you.
Thank you, Manju. Thank you, Manju, so much. And I am so proud of you, for initiating this particular series. And I think the students will get really benefited because I have learned a lot from others' experience and my mentors. And I think this is a wonderful program. So thank you for organizing it and inviting me. And thank you, Dr. Ramakrishnan, for your support for this program. And thank you to the whole department, I'm sure, must be involved in such initiative. I want to start with thanking all the students, for joining me on a Saturday evening. I'm sure your lab experiments must be running. There are a lot of family needs that you might have to take care of. But thank you for joining me today to learn about my journey. And I hope this particular talk could help you in carving your own future. So here I will share my slides, and I am actually, after my talk, I'm actually looking forward to a lot of discussion with you guys, answering any questions you guys have. So here goes my slide, and let's see. Can you guys see the slide?
Hello.
Can you guys see my slides?
Yes, Dr. Gandhi, it's visible.
Awesome. So as Dr. Ramakrishnan said, success is a journey, not a destination. And this is what I want to highlight with you, like.
Yeah, it's in the sense we need not worry about more if more people join then.
Dr. Manju Jain, can you mute your microphone, please?
Okay. I think Manju was on a call. So success is a journey, not a destination. And I think we have to remember it's an important point. So you guys must have, your next step sorted out. What is the next step? But I would say, that next step is not all your life. So as you go to your next step, you will find that your interest and your passions might change, or even, you might think that, okay, maybe I want to try a new career. I want to learn about a new thing. Career is a vast, there are so many opportunities available for all of us, but sometimes we don't think about all those possibilities available to us. So hopefully during this talk, I will reflect on my journey and you could maybe learn some pieces from that. So in this particular talk, I'm going to talk about in 3 different sections. So first section, I'm telling you, what has inspired me to be who I am. In second section, I want to share some of my academic research that I did, and I will promise you that I'll keep it high level. And if I don't keep it high level, please ask me a lot of questions after the talk. And in the third sections, I want to bring to you some of the learnings that I had during my career. So many years ago, I was born in this beautiful India, and I'm very proud to be born there. And I know India is at a very different level and a very different mindset right now. But, in my childhood, the main trouble for girls born in India was they were like a burden for the society. They were burden for family. And, in my family, we were 3 sisters. And, when my third sister was born, my whole family was like kind of in a shock, or in a kind of sadness that, there is another girl. And there were a lot of talks in my family for exchanging my sister for another boy or something. And that made me think, why girls are not appreciated, why girls can't do what boys can do. And it still resonates with me a lot. I feel very hurt by that concept during that time, and I'm sure India is at a very different level at that time. And I think one thing that I learned, that why girls was considered as a burden because of all the effort parents have to put for the girls' wedding. That one night wedding day, how much money parents have to put, and then they let the girl go into another family, then she's not part of your family, she's another family. And I have very different views about that, and I feel like girls can do as much as boys can do. And I know now society is at a different level, and there are so many programs encouraging women, but I am wishing and I'm hoping that you guys are at a stage where the girls are as much respected, and their talents are appreciated as much as boys. And I'm sure the society is at a different level. But this is what has inspired me to be something, because I did not want to be a burden on my family or burden on my husband. I wanted to see that girls with their hard work and with their passion can do as much as anybody else can do. So I want to thank my parents. When those times, it was not easy for my parents to think out of the box. They have let me do my PhD, and so many family members, I know my grandparents were very upset with my mom and my dad that they are not marrying me early in my life, and that all the marriage availabilities will be shut down once I'll finish my PhD. But my parents believed in me. They gave me the opportunity to study and to be who I am, and also to my sisters. So I'm very thankful and being very lucky to have them in my life. As Manju said, I did my PhD from National Institute of Immunology with an amazing mentor. And I think who I am today is because, what he taught me about science, about being honest, about being transparent, and how to do science in an honest way, and how to think about patients. So I did my PhD studying B-cell biology in lupus patient, and my only aim was, what is wrong with these autoimmune diseases? Maybe understanding of that could help us to really find treatments for them. And I am very sad to hear, there are so much researches going on, but still in a lot of neurologic diseases and autoimmune diseases, we still don't have a good target. But, we have a lot more understanding, how things were like 20 years back when I did my PhD. We are in a very different science era. And in my next projects, I think I'm coming back to lupus biology. So I think it's very interesting to understand these autoimmune disease and find new targets from that understanding that could really help patients. And here in this slide, I'm showing one way, because after I did my PhD, my only thought was I have to go for postdoc fellowship in US in a good college, because that's what most of the PhD students from my institutes were doing. And I think Manju knows that experience as well. A lot of people who study research at National Institute of Immunology are encouraged to go for a postdoc fellowship. And I think that one path actually opens up many, many different paths for us. But at that time, the only thing I knew that I have to do a postdoc fellow. So I came to Harvard Medical School to work on another autoimmune disease, but which was also neurologic disease, which is called multiple sclerosis. And I'll talk, in the next few slides about what that disease is and what I have done in that particular disease. So it was not easy coming from India at that time, being a very pampered girl and coming to Harvard Medical School in Boston. Now we are very much connected by Skype and WhatsApp. At that time, we didn't have that many options to connect with your family. So it was a hard decision, and to be honest with you, it was not easy. It took me, I think, almost 3-4 years to really make Boston as my home, and felt like, okay, I belong here now. So when you will go to your next level, be prepared for that. It's not going to be easy, but I'm sure it will be worth it. And in next few slides, I want to share with you some of the research that I've done during my academic career, and I'm only picking up 3 projects, not to bore you on a Saturday evening, and I'll try to keep it very high level. So during my academic career, I worked on, I think, maybe 15 to 20 different types of projects. I had about 45 different publications, peer-reviewed publications, presented at various national and international conferences. I was a reviewer for multiple manuscript or journals, and also reviewer for some of the grants for MS Society and for NIH. So the first topic I want to talk to you about, I worked during early career in academia, was to understand the immune system biology. So I did some work on T cells, regulatory T cells, dendritic cells, monocytes from multiple sclerosis patients, and understand how these cells are different in multiple sclerosis patients compared to healthy controls. To my surprise, I found that, all these markers, the main trouble in all these autoimmune diseases is, we don't have black and white signature. If we understand what particular cell goes wrong in autoimmune or neurologic disease, we might have an easy target to address. But I think there is, in these autoimmune and neurologic disease, there is spectrum of things that goes wrong. And I think that's where we want to take science, is to understand that particular spectrum, and that spectrum of what is altered in the disease versus health is very diverse. It can be very dependent upon a patient, and that's what I think right now we are working on, is the personalized treatment. So in this particular project, I worked on regulatory T cells and understanding what induce these T cells. So I worked for inducing the regulatory T cells, and this was mostly on all human T cells. This work was inspired by one of my colleagues, Francisco Quintana, who has done a lot of work in the mice biology, and we collaborated to understand the importance of this pathway in inducing regulatory T cells. So AHR is a receptor that is present on liver, kidney, and lungs, and also a lot of immune cells. And this pathway is mostly involved with taking care of toxins in the body. But we found out that if you activate this particular pathway, we can induce regulatory T cells. And I keep saying the regulatory T cell. What are regulatory T cells? So regulatory T cells are kind of what I say is like cops for your immune system. And I do not know where you are in terms of your understanding of immunology. Many of you already might know about these cells, and some of you might not have heard about it. But these particular cells are like cops, and they keep your immune system in control. So as you know, whenever there is a virus or a bacteria attack,
so Rupali, you may please, sorry to interrupt, you can click the stop, the hide thing, so that we see the details of your slides.
Oh, you couldn't see, sorry.
It was just a little thing at the bottom, but then yeah.
Oh, okay, sure.
Please.
So, awesome. Everything else is good, Manju? Yeah, okay. So these regulatory T cells are like cops. They keep the immune system in check. So whenever there is a pathogenic attack or a infection comes to your body, our immune system mounts an immune response. And so these regulatory T cells once the infection is gone, they tell the immune system, all is good, now calm down, we don't have to mediate an inflammatory response. So that's why you see here a balance that shows fire, which is, when immune system is active and mounting an immune response, and that these anti-inflammatory responses by regulatory T cells keep the balance in control for our immune system. So here, this is a lot of detail, lot of data here, but I don't want to go into too much detail. But I wanted to tell you that when we activated this particular AHR pathway on T cells, we generated TR1-like of immune cells. So these cells were the ones that were functioning through IL-10, which is one of the immunosuppressive cytokine, and also by engulfing the inflammatory cells or the dying cells. And when we activated this particular pathway in presence of another cytokine, we found that these particular cells become into FOXP3 cells, which are more like natural regulatory T cells, which are more commonly present in our immune system. And maybe, you guys might have heard about the FOXP3 regulatory T cells, which are very popular cells in human as well as mice biology. So this particular project, I don't want to go into too much detail, but what I said in summary is that, when you activate this particular pathway one way, it induced one type of regulatory cells, but if you add on some other cytokine mix, these cells become another kind of regulatory cells, and they function in a different way. And we also try to understand what are the upstream molecular pathway that was involved in generation of these cells. And I'm very thankful that actually this particular study, we published a lot of papers. I think I was involved in 2-3 of them in the human biology, but my colleague had continued this work in mice biology. And I know they are now testing, if we can induce some of these cells to really help in the autoimmune diseases such as diabetes and other autoimmune disease, to generate some of these cells in vitro and then take them to treat patients who are suffering with some of the autoimmune diseases. Okay. The next project, will need some understanding about what multiple sclerosis is. So multiple sclerosis is an autoimmune neurologic disease. So what happens, the multiple sclerosis means there are lesions in patient's brain. That's why the name comes as multiple sclerosis. And this particular disease, have different trajectories. Patients have different types of disease experience. Some of them have relapsing remitting course, which is shown in the blue line here. Patients have disease onset and then patients get better. As you see the arrows going up, the line going up means the patient are having the disease and then they get better. In the primary progressive, which only present in 10% of the patient, these patients start to get worse right from the beginning. And the patients have the disease and then they continue to get worse. And we don't have, I think we have now one treatment in the market for last 3-4 years, but there was no treatment for this disease for many, many years. Going back to relapsing remitting patients, most of the patient, about 90% of the patients, start the disease as a relapsing remitting course. And when 15 years back when I started to work on multiple sclerosis, there were only 4-5 treatments available, but today we have more than 20 treatments available for multiple sclerosis, but most of them address the relapses of the disease and not the progressive part of the disease. So now the biology is to understand what causes the disease progression and how to inhibit that progression or take care of that progression. So about 50% of the relapsing remitting patient becomes secondary progressive. So these green dotted line, as you can see here, shows that these patients have relapses during early on in their disease course, but then they become worse on a continuous basis, and that's why these patients then are called progressive patient, and this disease state is secondary progressive disease. So, this particular study that I worked on took, I think I was working on that particular study for 7-8 years. And I think this was very important for me because I was thinking, if we understand the biomarkers for these neurologic diseases, that can help us classifying the patient in a better way so we can treat these patients better. Going back to the personalized treatment. So understanding the patient more based upon the biomarkers so we can find the right treatment for the patient. And some of the uses of biomarkers are, to early diagnose the patient, because if you catch the disease early on, you treat the disease early on, you can find, maybe you can treat the patient better. A prediction of the disease outcome. If I know that a particular patient is going to progress later on in the disease course, maybe I will choose a different treatment for these patients early on so that the patient could benefit and can live a better life. These biomarkers also help us to measure the disease activity. If I know the patient is going through relapse and if I can treat the patient, maybe patient's disease progression will come later on, and maybe patients might not have a disease progression later on. And also, to understand, we need the treatment outcomes, right? If I have a patient being treated on one particular drug, but for 6 months I am not able to figure out whether the patient is getting benefited by the treatment or not, then it's very hard for the patient, because for 6 months the patient is just suffering. So if I have a biomarker that can help me tell that patient is actually benefiting from the treatment, then I can make better choices for the patients early on. So that's what I think the biomarker research was very important for me, to understand the patient biology so we can treat the patient in a better way to help patients live a better life. So our objective was to find different biomarkers to identify the disease subtypes, predict the disease worsening, and to identify the treatment response. So the biomarker that I focused, one of the biomarker, I worked on several biomarkers along with my colleagues who had different expertise, but one biomarker that was very hot in 2006, 2010 was microRNA. So microRNAs are small RNA molecules that actually binds to the RNA molecule and tells this RNA molecule to degrade. And in 2006-07, there were several studies that showed the importance of these microRNAs as biomarkers, and they can be measured in serum and plasma from the patient. So we looked at this microRNA biology to understand the disease, MS, multiple sclerosis specific biomarkers for patients. So this particular slide shows that biomarker study is not one project that you do. So it is like a commitment, like a lifelong study. So it's just like a clinical trial, where initially you do some discoveries, like how did we do discoveries in the lab, then you have to confirm those discoveries at multiple level, and that's what is shown here as validation 1 and validation 2. And then once you validate in a significant number of patients, you have to develop the assay so it could be used in any patients all over the world, and in a very fast way and in a very easy way, right? And if you think about these biomarkers, we know that there are biomarkers for diabetes, and how this insulin test is so helpful for patients to know about the diabetes, right? But for a lot of autoimmune and neurologic disease, we lack these biomarkers, and we need to spend a lot of resources, time, and have a lot of patients to really develop, significant biomarkers in these areas as well. So this was the different steps that I took for my microRNA biomarker study, and some of them I'm showing here. Maybe we can have a quiz at the end of the talk, and I'll quiz, what are the biomarkers that we found out in microRNA based biomarker for different disease. And this particular slide shows how we found biomarkers which could differentiate a multiple sclerosis patient from a healthy control, which could differentiate a progressive patient from a relapsing patient. And we also found some biomarker that were related to the disease activity, which was EDSS is one of the measure for multiple sclerosis patient, and we found several microRNA biomarker which could relate with the disease in MS patients. And here is the validation study. Once we discovered the biomarker, we confirmed it in about 100 patients in our cohort. Then we also collaborated with multiple cohorts. There were one cohort in USA, one cohort in Lebanon, and one cohort in Sweden, and tested these biomarkers again so that we can confirm that these biomarkers were not specific for Brigham and Wound patient. Worldwide, wherever we go, we can check some of these biomarkers and could check in different types of patients and from different ethnicity and different geographies, and can confirm that these biomarkers are good tool to develop further for the clinical testing. This particular study led to many different studies. We tested biomarkers in many other diseases beside multiple sclerosis. We checked biomarkers in ALS patients. We also checked biomarker in other neurologic disease, and some of that work was actually not published as well because of my decision to move from academia to industry. We found that these biomarkers are related to some of the MRI outcomes in neurologic disease. We found how microRNAs in the stool could help to also differentiate a multiple sclerosis patient from a healthy control or other neurologic disease patients. And we also found these microRNA, how to develop them as a more detailed biomarker. I worked with 5-6 different institutes across USA. They were also part of the same funding that I had from NIH to really understand this microRNA biology, and then to define the test that we could use to identify these microRNAs in serum and plasma. So that was a very, very exciting project for me. And I'm actually looking back, I'm very proud of these particular studies and the collaboration I had with other colleagues for this. One other study, and I think was very novel that I loved, was understanding the gut microbiome. That idea of working with the stool samples disgusted me when I started this particular study. But then, I understand, gut microbiome is actually very, very important in understanding various diseases, because, we all are inhabitant with millions and trillions of bacteria in our gut. And that bacteria is actually help us digest our food and live a healthy life. And if there are changes in that gut microbiome, that could lead to diseases or unhealthy states. And now, when I started in 2010, there were not a lot of studies to work looking at the gut microbiome, but now we understand various assays that we could use to understand this gut microbiome. And there are several studies going on in various diseases to understand gut microbiome and how they are changed in disease compared to health. So this particular study involved about 100 patients, and this diagram shows what we did. Besides testing the gut microbiome, we also tried to do some serum analysis and some cellular analysis just to show that if this gut microbiome is changing, it's actually affecting the immune system. It was changing the immune system. And here is a slide that shows the data, and lot of colors, lot of information. You don't have to remember that, but just wanted to show, kind of give you a feeling that when we tested MS patient, multiple sclerosis patient who were treated or not treated, or MS patients, multiple sclerosis patient compared to control, there were different phyla and different genera of bacteria present in their gut compared to healthy controls, or compared treated patients to an untreated patient. So these graphs actually shows what different bacteria or their genera or their phylum were changing in disease compared to healthy controls. And we tested it using 2 different technologies. At that time, we were not sure which one of them is better, because that was very early on in the microbiota science. So we tested during 2 different tests, and we got similar results, which encouraged us to show that whatever we were doing, we were on the right path. It was not a false signature. And this particular study, we looked at the microRNA also in the stool samples to confirm, what we found in the serum in the previous project that I was talking about. And then also one of my colleagues did a probiotic study, where she fed the probiotic, which we know are the good bacteria, to multiple sclerosis patient, and see, how that if that can help the patient feel better. And that was a very small group of patients, but it seems like there was some benefit of treating the patients with probiotic in multiple sclerosis. That was supported by the study. Beside these human studies, I know there were a lot of mouse studies that my mentor continued to do, taking the bacteria and putting them in mice and seeing, if mice disease get worse or better, treating with that particular bacteria. So it was very exciting project, and, very innovative and novel at that time. And I loved working on these projects. So, once from spending like 13, 14 years in academia, doing multiple different research and working, like I said, working on multiple different projects, I wanted to do something different. As you grow, like I said, your passion change, your interest change. And my whole, I understood, like in 2017, and I was always feeling I want to work for patients, and that's why I was doing the projects, I was doing research that can benefit the patient in some way. And as I was doing those research projects, I was collaborating with a lot of industry here in Boston, and I found out that a lot of my research interests are actually matching with what industry is trying to do. So I thought that why don't I try and make this jump from academia to industry to explore how can I help patients in a different way. It was not an easy decision, because I spent all my career to be an assistant professor, to be a professor at a university, and I was very doubtful of my decision that am I making the right step? Is this the right step? Because 20 years I've done one kind of thing, and now changing my path, is it going to take me in the right direction? But I think one thing that I kept in mind, I want to work for patients. I want to build science that can help patients, that can cure diseases. And that passion actually took me in the right path. And now being the medical director, which is part of the medical affair at Sanofi Genzyme, my major role is to provide information to physicians so they can make the right choice for the patient. And that actually fulfills me a lot, thinking that I'm working for patients, I'm working for science and doing the right thing for the patient and for the doctors. Being part of medical affair, I learned that how PhDs could work in a very different way, coming, different from lab work. So I always thought that being a PhD, you can only do research, but I found out being PhD, you can do so many different things in industry. And I collaborate, being part of the medical affair with medical research, where we look for, what are the additional data available for the patient and how can we make our treatments better and more affordable for the patients. We work with medical science liaisons who actually deliver all the safety all the efficacy outcomes from our products to the doctors, so they can make the right treatment choice for their patients. We work with medical education, we work with medical communication, who help to train the team, both commercial team, the marketing team, which actually goes out and sell the treatment or the drugs, and also the medical team who actually goes out and talk to HCPs about the treatment goods and bads. So I think PhDs can, like for last 4 or 5 years, I learned that PhDs can do so many different things just beside doing the research work. And I'm happy, I'm sure there will be a lot of questions from this particular my role, but I'm happy to address them after my talk. So this is my journey so far. I did my PhD in 1998, joined the postdoc fellowship in 2004, became the instructor in 2008, where my role was not very much different, but, it was more permanent position in the academic career. Became the head of the biomarker around 2010, was managing the biorepository for 2,000 MS patients, collecting their serum, plasma, stool samples, TS samples, and everything from these patients so we can find the right biomarker, and also was leading a team of wonderful collaborators or colleagues who have expertise in identifying different types of biomarkers. Became assistant professor in 2013, and I think that was the day when I thought the dream come true, because all my life I thought that, okay, this is what I want to be.